Hello everyone, my name is Jaime and thank you for your interest in our last paper on allergy regarding the role of the ligand of plant lipid transfer proteins. What makes a protein become an allergen is still an unresolved question, and regarding this topic, within last years, attention is growing to the ligands that are transported by allergens. Previous work from our lab shows that different LTPs carry the same ligand once purified from natural source. In this work, we wanted to deepen in the role of the ligand in sensitization, and we asked ourselves if mice will be sensitized when the ligand was complex with proteins from other families. We chose 3 proteins, PRUP3 as LTP model and control, and 2 non-related proteins, VSA and OVA. By in silico and in vitro analysis, we confirmed that VSA and OVA were capable of binding the LTP ligand, thus forming a complex. The 3 complexes were used to sensitize mice. 2 strains were used. And in all cases, we found that only when the ligand was present, there was a drop in temperature, characteristic of anaphylaxis, as you can see in the bottom right graphs. Also, previous work from our lab showed that the LTP's ligand is able to be phosphorylated by human sphingosine kinase, and the resultant product mimics sphingosine 1-phosphate. To deepen in this role in sensitization, we produce an homologue of the ligand with similar chemical structure but lacking the radical susceptible of phosphorylation, and we call it non-P ligand. We compare both ligand and the non-phosphorylatable version on the VSA sensitization model, and we found that in the case of the non-phosphorylable version, there was no drop in temperature compared to the native ligand. Also, humoral response was analyzed as a ratio of IgG1 versus IgG2A, and as you can see in the top graph, the results show that the non-P ligand have a ratio similar to the control one. Finally, we also analyzed leukocyte infiltration at the place of sensitization, and as you can see in the right graphs, we found infiltration only in the case of the native ligand, infiltration that was not present in the case of the non-phosphorylable version. Given the mimicking properties of the LTP ligand to sphingosine 1-phosphate, we sought to analyze its capability to promote leukocyte migration. So, we incubated cells with VSA ligand or VSA non-P ligand, and the resultant media was used to analyze THP1 migration in the presence or absence of fingolimod, a well-known sphingosine 1 receptor inhibitor. The results show the migration of cells only in the case of media derived from VSA native ligand, and this migration was abolished or not present in the case of the non-phosphorylable version, confirming the role of phosphorylation in its mechanism of action. This work was done thanks to our collaborators and funding, and you might find more information in our lab webpage. Thank you for your time.